And
to
that's
hear.
okay, so I've turned on the audio.
Yes.
Okay.
Now, if you go over to this one.
Should I put the camera on?
Yeah, sure.
Okay.
And yes, and here if you do share screen, the application screen, and then you can choose which one.
This one, I think.
Brilliant.
Okay, let me just close that on this one. And then
Brilliant.
hopefully that works.
Fantastic. Great.
I'm not sure what the online people can see, but hopefully that's okay.
Yes, Mike can see. Oh, brilliant.
Okay.
Can you hear her as well?
Can you hear me?
I think maybe we can, in the chat perhaps I can. Oh, yes.
Okay.
Very sensitive, very sensitive. Okay, so maybe I can introduce you.
Okay, yeah, thank you.
Sorry. Hello and good afternoon, everyone. It's great to have Dr. Caroline Roney today from Queen Mary, London, and she's a lecturer there, and in computational medicine and medical and MRC skills development fellow. So her title of her talk today is predicting atrial fibrillation throughout treatment outcomes through computational modeling, signal processing, and machine learning. So very core to Insignio research focus. So please, Karen, whenever you're ready.
Thank you. Thank you very much, Panaki and Richard, for the invitation today. Yeah, and thank you all for coming and for attending online as well. Hopefully you can all hear me okay. So, great, so I'll just get onto the talk. So here's an outline of what I'll be talking about. So I'm going to start with some background and some research aims. I'll then go through how we make our patient-specific models. So a lot of the work's gone into developing this pipeline, so I'll be a component of the talk. And I'll then be able to go into some actual research, and I'll go through some case studies for how we essentially predict either acute outcome of the procedure, and we'll then move through to predicting clinical AF recurrence, so whether the disease recurs or not. So my research is focused on atrial fibrillation. So as a lot of you will already know, activation of the atria, it causes a rapid and irregular heartbeat. It affects a large number of people, and one of the key challenges with it is that there are multiple underlying mechanisms. So you can see lots of the different electrical types of wavefronts you can have here. And as well as this, there are lots of different structural features that affect the atrium and electrical changes. So it can be difficult to treat AF because it's challenging to personalise treatment. So what's our clinical motivation? So as we said, in specific patients, so ones who haven't had atrial fibrillation for very long, catheter ablation therapy, which is where regions of the atria are isolated or burnt. So the red areas here have been isolated to isolate the pulmonary veins. This can offer a highly effective treatment, but in patients who've had atrial fibrillation for a longer period of time, this has a much lower success rate. And this is partly because it's a large heterogeneity in patients, so it's challenging to personalised treatment approaches because you've got all of this heterogeneous information. So we're hoping to really use patient-specific simulations with some other approaches to say how should we use this heterogeneous information to personalised treatment approaches. So that's just a bit of clinical motivation. The data that we're working with, so we've got imaging data. This is often MRI data, it could be CT data, and we've also got electroanatomic mapping data, so electrical data that's collected during the procedure. So you've got the different catheters moving around here and collecting the electrical signals, and we can use these to personalise our simulations. Further challenge of what the clinical data, there are lots of different systems that can collect this data, lots of different catheters with different arrangements of electrodes, and we tried to bring all of this information together into our patient-specific models and try to then use this to understand the mechanisms for these arrhythmias. So we've covered the clinical problem. Now, what's the current state of the art? So first, we've got large clinical population data sets, and these can inform treatment approaches for generally the average patient within a population. We've also got increasingly more detailed patient-specific models. So these tend to have more and more detail, but they're not, a lot of the time, they're not really using the insight from the large population data sets. So the vision here of what we're working towards is to try and use this information together. So to try and use the information from the population studies to try and say how we should personalize our patient-specific models. To do this, we need large virtual cohorts that capture the real heterogeneity in the anatomy and the fibrosis distribution, the different features of the atrium. And currently these cohorts are small, so you may have, say, 50 patients, and we're now working towards having much larger cohorts of models. And the final challenge is that we typically often model what happens immediately after the procedure, so say in the 30 seconds after the ablation procedure, so the acute response. But what we'd actually like to know is what happens in, say, the year after the procedure. And going between these different timescales is very challenging. So our aims here really are to develop an automated tool for determining how to ablate from imaging data and electrical recordings, and the tool will use biophysical simulations with some machine learning techniques to try and predict the ablation outcomes. To achieve our aims, we're using a combined approach, so we're using some signal processing techniques, some mechanistic biophysical modelling techniques. So the signal processing I started working on during my PhD at Imperial. I then moved to Bordeaux and worked more on mechanistic biophysical modelling. We're now trying to combine this with more machine learning techniques and going towards larger populations. And ultimately, we'd like to translate this to the clinic. So, as I said, we want to use atrial modelling to understand the mechanisms and predict ablation outcome for using these patient-specific models. And we need to make them from readily available clinical data. So this brings us on to our pipeline for how do we make models. And we want this to be reproducible in general. So if we have the same data, we get the same model. Now, there are lots of steps involved in this, which I appreciate. There's, a lot on this slide, but essentially, we want to construct an anatomical model, calibrate the model to our electrical and structural data, initiate AF, and post-process the simulations, and perhaps simulate ablation. I'll go through each of these steps in turn and focus on some of our new developments. So first of all, how do we get an anatomy? So for this, we use the CemrgApp, which is developed mostly in Steve Nidra's lab at King's College. So here it will do automated segmentation or semi-automated segmentation. So this segmentation has actually been done by one of the clinical fellows. You've got the left atrium here, so the left atrial body's in red. You've got the veins the pulmonary veins are in green, and then the mitral valves in blue. And this is an open piece. An open source piece of software, so if you'd like to use it, have a look. And what we can do with this software is we can do some pre-procedural analysis, so that we can use to target patients, maybe say how to ablate them, perhaps make some predictions, and then also post-procedural analysis, so looking for gaps in ablation lesion sets on second imaging scans. So it just gives you an idea of what we can do with the software. And we're now moving towards more automated segmentation. So here it's using a unit approach to predict the left atrial blood pool segmentation. And we can then register that with the late gadolinium enhancement to get our fibrosis distribution. And this takes out a lot of the variability between users in the segmentation. We used to have a lot of different softwares for making our models, so a really large sequence of steps. We're now moving towards putting them all into CemrgApps. This is a lot of work that Jose's been doing in the King's College London group. And here the idea is to be able to clip the veins, to be able to make all the changes you need to make in your mesh, to add fibres to your mesh, all within a single piece of software. So that's what we're working towards. The different steps that we need for this. So once we've got our anatomy, we need to be able to register either electrical data to it, or we might want to register data between different patients. So that's why we developed this atrial coordinate system to deal with variability between different atria, so they can be quite variable in their morphology. And essentially they all get mapped just to a square in this case, using 2 coordinates. And this means that we can just do all of our analysis in a standardized way. We later then fixed some of these boundaries so that we map the pulmonary veins to the same location each time and the appendage to the same location to give us a more standardized mapping and improve our registration. Now, once we've got this mapping, we can then use it for other things. So atrial tissue, it's electrical anisotropy, and that's where conduction's faster along fibre directions than transverse to the fibre directions. And to include this in the models, we've constructed a human atrial fibre atlas, and we did this from this really nice data set from Natalia Trayanova's group and Elliot Murphy's group, and it has human ex vivo DT-MRI data for the atria, and we put all of these data sets into the atrial coordinates and registered them together. And you can see the 7 different data sets here. So you've got the left atrial endocardial surfaces at the top and the epicardial surfaces at the bottom. And we've got the equivalent for the right atrium as well. And you can see there's quite a lot of variability between these 7 cases. And then what we can do with this is register these all together using the atrial coordinates and calculate an average map. And we can then have a look at the effects of these different fibres on our model predictions, essentially, and to see which of the fibre maps give the most similar model predictions to the other fibre maps. Another factor that we need to include in the model is atrial fibrosis, and this has been shown to correlate with whether atrial fibrillation recurs or not. There are many different factors involved in fibrotic remodelling. So you have kind of an interstitial type fibrosis. You also have a replacement type fibrosis. You have slowing of conduction velocity, changes in the electrophysiology. You might have fibroblast coupling. So the first approaches that we took were essentially a kind of a scattergun approach. So to look at all different combinations of these, and then to see where we predict we have electrical areas of re-entry across the different models, to see whether we have common sites or not. What we're moving towards a little bit more now is to try and use data. So this was collected at St Thomas's Hospital. We've got paired data, so patients that have had both late gadolinium enhancement MRI data, where these higher intensities on the imaging are thought to represent more fibrotic remodeling areas. So we've got these data, and these patients also have electroanatomic mapping data. So they've also got the conduction velocity map. And so then we can then register these, again using the atrial coordinates, and look at the relationship between them. So far we've just done this for 10 patients, but this has given us a mapping, a preliminary mapping between these that we can use for our models where we don't have electrical data. And all of this analysis really needs the electroanatomic mapping toolbox, so OpenEP software, and this was developed by Stephen Williams at King's and Nick Linton at Imperial, and we've also have other people who are adding to it now. So the toolbox just gives you a standard framework for importing and post-processing the data from the different systems, and yeah, it's available open source again at OpenEP. So the next factor that we need to include is how do we calibrate the electrophysiology? So we know that there is electrical anisotropy, but this data is, so it's faster, electrical propagation is faster along the fibres than transverse, but these maps are often not available clinically. So what's happened here is one of the clinical fellows has paced in 3 different locations, so on the septum, at the right pulmonary vein, and at the coronary sinus, and we've got activation maps calculated from each of these directions. we can then use these conduction velocity maps and bring them together to give us an estimate of what we think the anisotropy looks like, so what do the average fibres look like, and extrapolate those. So this is a simple technique for doing this. We're now trying to move on and work more with incorporating the uncertainty in this, and working with Sam Coveney, who's here, and Richard as well, to try and bring this data together so that we have more of an estimate of the uncertainty on this and more advanced techniques for doing this, so that you don't necessarily need three directions. But here anyway, we've got some validations. This is rabbit optical mapping data. We've also got histology, and so we can try and validate the techniques. We've calculated this for 6 paroxysmal patients and made an atlas, 6 persistent AF patients. And of course, we can compare these to the DT-MRI data that we've got here. And as I said, the ways that we could do this now, we could include more of the uncertainty quantification for conduction velocity. I'll come onto this a bit more later, but again, the codes are available here. So now, how do we initiate atrial fibrillation? So because we spend so many hours essentially trying to initiate atrial fibrillation and failing, we've just come up with a very standard protocol for doing it. It's not the most physiological, but it means you can apply it across lots of different atria. It's very quick. So essentially, seed spiral waves in an arrangement that we want, and we calculate this just analytically using the atrial coordinate system, and we can then use this as initial conditions for generating our arrhythmia. So now that we've done our simulations, we can post-process the data. We usually do this using phase mapping. And then as a final step, we compare to clinical data. So this could be cardio insight data, it could be basket data, or it could be acutus data. So yeah, so lots of different data sets. So as I said, the steps, so we go from our imaging data, we make our simulation mesh, calibrate our data, start AF and run our simulations. So now that we've got our modelling pipeline, we can actually do some research. So our first studies aims to predict acute ablation outcome from our clinical AF recordings. So AF appears chaotic, and here we're trying to see whether there's actually more of a degree of order in the fibrillatory data if we analyse it probabilistically over time. And we want to see if we could use this to predict whether ablation works or not. So we were post-processing the Starlight study data set, and it's got basket data, so left and right atrial data collected simultaneously. And the aim here is to identify preferential activation pathways and predict ablation outcome. So we take the signals from the basket, and here we're using an optical flow type technique to essentially track the wavefronts over time. So it's like you could use it for tracking cars on the motorway. It's the same kind of technique. And we just do this for the wavefronts. And we can then calculate the average vector field over time, where the larger arrows tell us that our direction's repeated more often. And what we found was that cases for where this pulmonary vein isolation worked tended to have this flow, as you can see in the green boxes, from the pulmonary vein entry into the left atrial body. So yeah, so we saw this flow, and then cases where pulmonary vein isolation didn't work, we didn't see the same kind of flow. So what we then did was we did some simulations to test this and to do some validation and to look at the mechanisms for why this might happen. And essentially we just came up with a metric that that quantifies how much flow you've got from the pulmonary vein entry into the body. And this could represent if you had drivers in the pulmonary veins, or you've got a re-entry anchor in the pulmonary veins, then essentially it's telling you that it's more likely that the ablation approach will work. But you're actually only needing recordings in the left atrial body. So that's that has the advantage that you can measure it clinically, but it does need global recordings, and it also only works for this one type of ablation approach. So we're now going to use simulations to look at any ablation approach and to see how much data we actually require. And we'll then combine this with machine learning techniques to predict the acute ablation outcome. So as I said, so lots of different ablation approaches, which I'll go through. We'd like to be able to say what clinical metrics should you calculate for predicting outcome, and then how can we bring this together into a classifier so that we don't need to run all the simulations. So as I said, you could start with pulmonary vein isolation. So we've just kind of been through this a bit more in the last section of the talk. So if pulmonary vein isolation doesn't work, you might want to do something more like box isolation. So this isolates the roof of the atria, so using these blue lines here to isolate more of the tissue. And in this study, we were using simulations to go alongside a clinical study to see the effects of different boxes on this ablation. So they could only simulate one type of box, but we looked at the sensitivity of the size essentially. Another use of simulations here is that we can test lots of different ablation approaches. We can also try very extreme ablation approaches, obviously without harming a patient. So what we did here was we ablated the connections between the left and the right atrium, so Batman's bundle, the coronary sinus, and along the septal wall. And this represents a very extreme and speculative approach, but we could test it in the simulations and try to derive a metric to stratify patients. So this is just a use of potential use of simulations. And then we're excited about this study because it's a larger virtual cohort study, and we're finally using some machine learning with our simulations. So we had 50 patients with atrial fibrillation, 20 of them were paroxysmal AF, and 30 had persistent AF. We built simulations of them, so the modelling was here, and we then trained a classifier to try and predict the outcome of different approaches. So the approaches that we looked at, so as I've already spoken about, pulmonary vein isolation, another anatomical type, so box ablation, where you isolate the roof. We ablated areas of electrical re-entry, so these areas in grey here are the areas that are ablated, or areas of fibrotic tissue, so again, the grey areas here. And lots of this work was by my student, Marianne Beach. And what we saw here was that if we have our 50 different models, we see different outcomes depending on the model. So these models have different anatomies and different fibrosis distributions. The ones with the white background still have atrial fibrillation after the ablation, whereas the ones with grey have either terminated or it's a more simple re-entry. And we also see if we have the same model, but we apply different types of ablation, we see different outcomes. So in this case, the ones with more extensive ablation, so the box or all areas of fibrosis or all electrical areas, the arrhythmia is either terminating or becoming a more simple macro re-entry, whereas these cases are still sustained. And we could see that across the population of models, you have different ablation approaches are optimal for the different models. So what we then wanted to see was, could we just train a classifier using some metrics from the images or from the simulations before the ablation to predict the ablation outcome. And this plot is just a Shapley analysis, so it tells you the importance of the different inputs to the classifier. So the ones at the top have a larger effect on the classifier prediction. So in this case, the remaining electrical area, so essentially if you have more areas of re-entry that you don't ablate, it's more likely that the arrhythmia sustains after the ablation. So it's kind of what you'd expect, but it's just giving you an idea of how important each of these are and whether they increase the likelihood or not of the the arrhythmia terminating. So yeah, so this study just shows an application of not needing to run all of the simulations, but being able to predict the outcomes using a classifier. So now that we've looked at that, what we needed to say was, well, do these responses actually match the long-term clinical outcomes? And if they don't, then why not? How should we change our pipeline? Can we even use the models? So here, as I said, we want to go from the short-term response, so in 30 seconds after the procedure, to the longer term, so say a year after the ablation. And this is difficult as the patient can change a lot during that time, and we're not even necessarily certain that we're getting the acute response right. So trying to use the models to predict the longer term outcome is challenging. So what we did here is we have just 100 patients here, but it's again a step up in terms of how many patient-specific models we have. And for 34 of them, they had atrial fibrillation recurred after a 3-month blanking period, whereas for 65 of them, AF didn't recur. Unfortunately, one of the patients we didn't have the follow-up data for, but these are the ones that we've made the models for. So, yeah. So we then had to look at the clinical metrics to see if there's any difference clinically in the two cohorts, and we found that there wasn't. So these simple kind of clinical patient history metrics are not telling us whether AF recurs or not. So we then looked at the imaging data, and we actually did just a very simple analysis. So it could be that if you did a more complicated analysis of these images and processing of these images, you get a lot more information. But essentially, we took these images where the red and white areas are thought to have more fibrosis, the blue areas are thought to be more healthy, so it's late gadolinium enhancement data. And we just did a simple analysis of just the area of the tissue, the area of the pulmonary veins, the area of the fibrosis, and the area of fibrosis in the pulmonary veins, and we saw no difference between these metrics. So this simple analysis, again, isn't enough. We then looked at our models to see in the acute simulations, do they match the long-term outcome? And the models above the red line had no clinical AF recurrence, and the ones below had clinical AF recurrence. And what we'd like to have is that all the ones above the red line have a white background, so no atrial fibrillation in the simulations, and the ones below would have the grey background. But this isn't the case, so the acute simulation responses are not exactly matching the long-term response. So we need a new approach. So the approach that we're trying to use is to combine these simulations together with the patient history and imaging metrics to predict whether AF recurs or not, by training across the population of models. So as we kind of said from the first part of the talk, there are so many unknowns in the modelling pipeline. So we don't know how to model the fibrosis, how to model the fibres necessarily, what size the ablation should be, how to initiate the arrhythmia, and what the electrical properties should be. So the way that we've tried to address this in this approach is to create these simulation stress tests with different values for each of these, and then we take the outcome of all of these 11 simulations for each patient, and then use that to give us a patient-specific signature. So we're not expecting any one of the simulations to match the outcome, but maybe some measure of them will. So to give you an idea of what these might look like, so this is for one patient, we've got all of the different variations of the model. And we can see that in this case, this patient did have AF recurrence, and most of the setups we're still seeing a re-entry around after the ablation. So in this case, most of them are showing a re-entry afterwards. We then take all of this information together with the imaging data and the patient history, and we trained a classifier. And these plots tell you the results of the tenfold cross-validation. And we see that when we remove the simulations, so just the imaging and patient history, or just the patient history, we don't see a very good prediction at all. So the simulations are definitely helping us with our prediction. That's likely because you've got all the information on the distribution of fibrosis, and that's all brought together in the model. And we can also have a look at simulating more advanced ablation approaches to try and see if this will change the classifier prediction, and if this is more likely to work or not. So this is just a bit of a proof of concept. So that's so one of the main disadvantages so far of our large virtual patient cohort study is we didn't include personalised electrophysiology. So we had the personalisation from the fibrosis data, but we weren't including electroanatomic mapping data. And this is bringing us onto this case study where we just got some different approaches for looking at the effects of electrophysiology on where we see electrical reentries in the model. So the aims here is to look at the effects of conduction velocity, so how fast the electrical wavefront propagates, and the effective refractory period, so the minimum interval between the wavefronts, and where you see re-entry. And we want to test if we tune our models to these properties, do we see a better prediction? And we've got some different approaches for doing this, which I'll go through now. So the first study where we kind of looked at this was during my PhD, where we had optical mapping data from a canine atria, and you can see the re-entries going on here. Sorry, the video is a bit laggy. And then we used simulations to try and see if we could investigate different reasons for why we might see these areas of new wavefronts that we were seeing in the optical mapping data. So this is a way of combining the simulations with the experimental results, I guess, to try and investigate the mechanisms. We then looked at if we could parameterize conduction velocity. So when I was working at Lyric with Ed Vigmund, and this is just a way for bringing together how we could calibrate conduction velocity from CardioInsight data. And now, as I was saying, to try and derive a mapping between these metrics, so between the conduction velocity and the late gadolinium enhancement intensities, we've done this kind of paired analysis for 10 patients to give us a mapping. But this is just for different bins of data, because we've only done this for 10 patients at the moment, so we can't really do much more than that at the moment. So we then took this mapping and calibrated patient-specific models. We've got ones without calibration at the top, and then one with calibration at the bottom. If there's not much fibrosis, there's not much difference in the simulations. But in cases where there's more fibrosis, we see more differences in the simulations. So kind of it has more of an impact. And we're now trying to extend this to a larger cohort. So this is work as well with Sam, who's here. So we're looking at different methods for how to calculate conduction velocity for paroxysmal AF patients and for persistent AF patients. This is across a cohort, and this will give us an idea of how much these properties actually vary between patients, and how we can then include this data in the models. So one of the big challenges is how do we validate our modelling pipeline? And that's because we often don't actually know where areas of re-entry are. So for this, we're working with Sanjeev Narayan at Stanford, and he has a really large data set of basket data. So you've got kind of global recordings, and this can give us measures of the conduction velocity, of where the electrical re-entries may be, and the properties during AF. So the way we're approaching this is we've got basket electroanatomic mapping data. We actually also have CT data, so these anatomies will actually be CT going forward. But at the moment, we've got fibres, we calibrate our models. I'll come onto this more in a minute, but we interpolate the effective refractory period and conduction velocity, simulate AF, and then we simulate the same ablation that was applied in the clinic and see if we see similar features or not. Now we really need, sorry Sam again, so we need a way of calibrating our models to the conduction velocity measurements that we're getting during atrial fibrillation. So a challenge with this is you've got changing wavefront directions, you've got the catheter moving around. It's very difficult to assign activation times because the electrograms are very fractionated. So we've got lots of difficulties with that, and a way that we want to then process the data is to use uncertainty quantification. So we've got some really nice techniques developed by Sam. Again, it's on his GitHub here. But once we've then got our estimates of conduction velocity across, say, 60 seconds of AF, we can then give us an estimate of the mean, how uncertain we are in these metrics. These are actually using an older version of the code, so these look nicer now, but these are giving you an estimate of what the conduction velocity is, what the AF cycle length is. We can then use these to calibrate the models. And whether we calibrate these to the mean or to samples, so what we're trying to do now is to have a look at the effects of that on what the model predictions are. But essentially, if you see here, this is the calibrated model. We can try and compare to the clinical areas where they've seen re-entries indicated by these white boxes. and we can try and quantify this, apply the same ablation approaches. And what we found was that if we just apply pulmonary vein isolation, the AF doesn't terminate. But if we then apply the same lesions that they applied in the clinic, we did see a conversion to atrial tachycardia. But what we're doing now is extending this to more cases and seeing if we can use it to get an estimate of how much these properties affect the model predictions. So now for the final case study. So this is aimed at addressing the challenge that computational modelling takes a long time, and we need to be able to make our predictions on clinical timescales. So here we're using biophysical simulations with deep learning techniques. So the idea here is we use our modelling pipelines. So as we've spoken about, the different anatomies, different electrical properties for our calibration, and different fibrosis properties. We can have lots of different permutations of these, run simulations to predict where we have electrical reentries or phase singularities, and we can predict these before and after the ablation to also try and predict whether the ablation works. And the idea then is then to take these just as inputs in the atrial coordinates, and then try and predict the output phase singularity maps. And the idea here is that it will learn the physics. So these will be constrained by the physics and physiology that's included in the models. You then include more clinical data to just train to that to try and see essentially the features that aren't captured by the model, and to try and increase that complexity. So we've just started here with a very simple toy problem, so to look at the effects of anatomy and fibrosis and phase singularity maps. So we've got different anatomies here, different fibrosis maps, and then depending on these combinations, we'll get different predictions of the electrical driver locations. And we want to be able to go from the fibrosis maps to where we predict the phase singularities are. And this will be trained to where the biophysical simulation model predicts the phase singularities are. And we started just by using a unit approach that you'd use, say, for segmentation. So the idea would be to kind of try and somehow segment the imaging data to say where these phase singularities are. But the problem that we're finding is that essentially the model often over predicts these areas, and that's partly because as well we looked at lots of different permutations of the different fibrosis maps, different permutations of the anatomy, different permutations of the fibre maps, and essentially depending on the exact setup, you get actually very different phase singularity maps. So it's very sensitive to these initial conditions. So it's actually very difficult to predict. So we tried instead to see if we could predict it using slices of the imaging data. So we used the input slices and then tried to predict the areas of phase singularities, so like the red areas here in the slices. And again, the model over predicts. So we're trying to see now if we have more simulations perhaps, and we're also moving towards instead just trying to predict that overall whether the ablation works or not, rather than the exact phase singularity map, because that's very, very sensitive to all of the setup of the model, which I guess makes sense in that the fibrillation also is very chaotic. If you calculate a phase singularity map through a clinical recording, that can also change over time. So it's a challenging thing to predict. Now just some kind of areas of where we kind of go into now. So I think as a field, we're trying to share more of our tools and codes. So again, Sam's code here is a really nice example of that. We also tried to put some of our fibre maps online. There's OpenEP available if you want to analyse electronotope mapping data. And again, the CemrgApp. OpenCarp is the simulation software that we often use in our field, so developed by Ed Figmund and Gernot Planck and the Karlsruhe team of Axel and Olaf, so, and lots of other people too. So, but anyway, this is really useful for us as a field, because it means we can all run our simulations, we can share our parameter files, we can hopefully share our models, and it makes them more reproducible as well. So this is kind of areas I think we need to be moving towards, perhaps. As I said, yeah, these 2 open science platforms. The links are here, and like the next steps of where I'm looking to go towards. So I'd like to be able to update the models time in a more digital twin approach. So we've got scans at different time points after the ablation, and we're trying to essentially try and predict how the scan will change over time, so that we don't necessarily need all of those scans, and then rerun the simulations with these predicted data, so like a digital twin kind of approach. We'd like to be able to say what's the minimal ablation lesion that you need. So perhaps combining this with like a network theory type approach to find what lesions to use, to extend to antiarrhythmic drugs. We like to use UK biobank data. These are obviously lots of different areas, but just areas to go into. Perhaps more uncertainty quantification to say where we should take more measurements, and ultimately a clinical tool. So obviously this is a work of a lot of people, so I'd like to acknowledge the Cardiac Electromechanics Research Group, so Steve Nieder and Martin Bishop's group, all the clinical teams there as well, my PhD lab, postdoc lab, the Sheffield Leeds group here for uncertainty quantification, the DT-MRI data, Stanford collaborations, yeah, and my MRC funding, and just welcome any questions. Thank you for listening.
Thank you, Caroline. I have put in a chat message for those who are online to ask questions in the chat box. Apart from that, but very interesting talk. I was actually going to ask a question about biobank. So you said that you would like to utilize biobank data. Is there already a protocol for data collection with biobank? Are they already doing it, or is this something that you work?
I think it's for the atrial imaging is a little bit more challenging than for ventricular imaging. There isn't always all of the information, and you might need to reconstruct it more from the data that's there. But there is already a lot of data. There's also a lot of the genetic information. So I'm hoping to work more, like now I'm moving to Queen Mary with the electrogenomics group there. So they're working a lot on UK biobank data. So yeah, working more with them on that. So, but that's very early in that. So
thank you so much. Are there questions here? Yes, Richard.
Hey, Richard.
Well, lots of technical questions which we can talk about.
Yeah.
I was wondering what type of classifiers you've been using for the machine learning.
So yeah, so the classifiers we use for the machine learning. So there were we've just used simple ones at the moment. So we just compared logistic regressions, support vector machine, K and nearest neighbours. We haven't done, there wasn't anything much more complicated than that. At the moment, that's partly like that was quite a small data set. I think for the deep learning parts, whereas we scale up to more data, then we'll be able to do more. But
So did you have a favourite classifier?
Yeah, and for the data I showed for each, so they were different. The ones that came out optimal were different depending on whether it was the simulation model, the imaging, or the patient history. But they were all very close, to be honest. There was more difference, obviously, between which of the data sets you used and which of the classifiers we found, at least.
I will continue. Amazing talk, first of all.
Thank you.
I will continue in the same topic as Professor Richard, and I will ask about the classifiers. You mentioned 3 cases. The one was that the best approach was the one with simulation data inside. All these results, I'm guessing that is the same classifier. Am I right? You use the same, for example, SPM, and this is the output.
No. For each one of those, it shows the best classifier for the data set.
But I think this is not. But you don't compare this, because you cannot conclude if you compare different classifiers. This is no sense from perspective of classification, because every classifier is different. You cannot say that the simulation that has inside the synthetic data and the ordinary data, the best result is SVM, for example, and is this. And if we don't have the simulation, the best result is for regression, and this. So we need the simulation. You cannot conclude that because the structure, the model are different.
Well, I mean, kids pick. I mean, we have the results for the same classifier for each. It's just that they'd be there. Yeah, it always follows the same the same pattern. It's just that I presented the results in the figure that's the best for each. But yeah, you're right. Maybe it would be better to have it as a multi-figure where we have support vector machine at the top, for example.
Because to conclude about the input cohort that you use in the machine learning procedure, you have to have the same structure. If you change the structure, you create another.
Yeah, that's a very good point. So the pattern was the same.
Yeah, you had this, but you just conclude and say this is the best result. Makes sense.
Yeah, but yeah, maybe for presentation it would be better. So yeah, for the paper to include it that way as well. Thank you.
I have a lot of questions about deep learning, but I will not continue with it. An amazing presentation.
Thank you.
I'm waiting for questions from the online audience, but I have another question. So, in terms of using this method in the clinical pathway, of course, existing clinical pathways, and has anybody done a cost-effectiveness analysis of what would be whether this would how cost-effective?
Oh, that's a really good question. I don't know. I haven't seen anyone with a cost-effectiveness of including simulation. I mean, perhaps in grant proposals, people include that.
I mean, one argument
I don't know.
we've used in a grant proposal was that if you reduce the number of patients who have to come in for a repeat procedure, there's a cost saving there, but putting a figure on it is really hard.
Yeah.
Because it goes through the quality of life and all those surgeries.
Yeah, it's a very good point though. That would be an interesting analysis, I think.
Yeah.
So at one point you were using the UNet to predict phase singularities, and you were saying that currently you're sort of over predicting how often they occur, but it seemed to predict some of them well, but then it predicted a bunch of others that weren't there. Is that prediction though weighted for like, number of times that phase singularity occurred in a certain location, or was it just binary?
No, so it will, because it's the density map. So you'll more or less see everywhere that we've had a phase singularity, and then I've thresholded that. So yeah. I mean, it could be. I think part of the problem was that because we had lots of different permutations of the fibrosis map. So say if we had a slightly different permutation of fibrosis and anatomy, then the phase singularities would go in a different area of the atrium, and so it would also be predicting that area, even though that's not necessarily seen in this simulation, but it's seen in with this fibrosis map with a slightly different anatomy. We found that was a real challenge.
You had the fibrosis map, but you had it in your atrial coordinates, and so in theory, that the idea is that those are equivalent shapes, even though they're from different atriums. The idea is that you're in the same system, but then you said you're still seeing that the particular anatomy itself.
Because the simulations are very sensitive to it. Which is a challenge. And another thing is when we first did this analysis, we were using the old version of the atrial coordinates where we hadn't fixed the locations of the pulmonary veins. That was also including quite a lot of distortion. I think we will redo it again with with that fixed and see.
I guess it'd be interesting to try to keep more of the original topology in there.
I mean, there's
that can't really be put into 2D.
Yeah, I mean, there are. I guess there's more like graph-based, like neural network type approaches. Could be that those are more useful, but then you also still do want information on where you are on the atrial anatomy. So
you could add a couple of higher dimensions to that instead of just 2. You could have 7 coordinates.
Yeah.
It's just a thought because you're squashing different topologies into the same
Yeah.
2 by 2?
Yeah, so we have a distance metric, which is the distance between nodes, but I don't know that that's necessarily, so in areas where it's more distorted, it will be higher, but I don't know if that's necessarily the best way of doing it. It's a very simple way of doing it.
Third axis into it.
Yeah.
Okay, thanks. That's interesting.
Oh, thanks.
There is a question online from Patricia Martinez. The question is, she compliments the presentation. It's a very nice presentation. And could we develop an ablation planning tool with information coming only from the left atrium?
Yeah.
What do you think about the influence of the right atrium?
Yeah, I think Patricia is very right. I think we do need biatrial models. So I think a lot of the work in our group has worked on segmenting the left atrium, which is why we tend to work with left atrial models only. And it's more complicated to work out how to put the atrial connections between the left and the right atrium and how exactly to model those. So we've only done a small number of biatrial models, but when we did do a study where we had biatrial models, we found that some of the properties in the right atrium, so how many facing lobes she has in the right atrium, was important for predicting re-entry. So it's definitely important, and the literature's pointing towards that. So I think going forwards, we'll either use patient-specific right atriums, or we might move a bit more towards using like the statistical models like developed by the group that Patricia's in. So the Carlsruhe team have like given us a statistical approach for this is what the right atrium might look like, and go that way. So depending on how many what coverage we have of the right atrium in the scans. So but thank you.
I have one more question. So there's so many inputs that go into the model, right? Is it possible to do a backward analysis, trying to figure out which kind of pathologies, at least or maybe stratify all the patients into different kinds of pathologies, and from there try to figure out which inputs they would really need and precisely to be known to be able to stratify.
Yeah, I think it's a really nice idea, because at the moment a lot of the time we just have, if a patient has paroxysmal AF or persistent AF, but that's a bit of an arbitrary definition, and I think that you could do a lot more than that, and hopefully with the biobank data as well, it might be that we can say more with different genetic factors, for example, then we'll have subpopulations. But I think we're quite away from getting to that, but that would be really nice.
Mm-hmm.
Yeah.
Yeah.
Thank you. Thank you.
Okay, there are no more questions online, so thank you very much.
No, thank you very much.
Really nice.
Yes, thank you. Thank you. Thank you. Thanks, everyone.
Thank you. I close the online session. Thanks all the online attendees. Bye. Have a nice weekend.
Bye. Thank you.